I'll just start by setting the stage with the problem that led to the foundation of Open Targets, which is, of course, very familiar to everyone here, which is that drug development is a failure-prone process. There are various examples of this. This is one from a couple of years ago, where if you ask at each phase of taking a candidate towards an eventually approved medicine, what's the chance that it fails? And then when you multiply all those probabilities together, you get that right-hand side, which shows that cumulatively, maybe as much as 85% or perhaps even more of new candidates don't make it to drugs. And even more worrying is that this blue bit of the late phase 3 and approval stage are failures in those stages due to lack of efficacy. So even when we get to the most expensive part of the trial process, there are many cases where the drugs simply don't provide any therapeutic benefit to patients. And so Open Targets is put together to think about that problem with one specific, at least partial solution. And that solution really can be summarized by these data that were published by Matt Nelson and colleagues in GSK last year, which is they looked retrospectively at drugs which had gone on to become successfully approved, and considering the targets of those drugs and whether there was any genetic evidence in support of those targets. And the top line is using OMIM or Mendelian genetic evidence, where both the strength of the evidence and the specificity to the gene tend to be quite strong. And the second line uses so-called genome-wide association study type evidence, where often the individual effects are very modest, and it's not quite so straightforward to map a signal to the gene that's actually underlying that signal. But in either case, in the case of the Mendelian evidence, there's something like a 7- or 8-fold, and even for the GWAS evidence, nearly a 3-fold increased probability of success, given that the target had genetic evidence. Now, the question for Open Targets is, can we take this nice retrospective observation that genetics is useful in highlighting good targets and use it prospectively to feed into drug discovery programs? I'll just briefly, for those of you who maybe haven't thought about this before, introduce the idea of genome-wide association studies, a study design which was really pioneered about 10 years ago and has become one of the workhorses of trying to understand the genetic basis of complex diseases. Essentially, the focus is on collecting as large a set of samples as possible. So we might imagine the orange dots are individuals with a particular disease, and the purple dots are healthy controls. And the key insight was that complex diseases have many, many genetic underpinnings. Each contributes only a small amount of risk. And so identifying them requires not just a few hundred or indeed a few thousand, but often tens of thousands of individuals. And so recruiting very large sample sizes is a key to the success of genome-wide association studies. These individuals are then typically genotyped on these arrays that survey the common variation in the genome. So it's not at the level of sequencing the entire genome to look at every personal variant that individual has, but to look for the differences in frequencies of variants that are maybe present in 20 or 30% of individuals. And then you essentially do a fairly straightforward set of association statistics at every commonly varying position in the genome and ask, is this variant more common or less common in cases compared to controls? You get a picture that looks like that middle blue diagram where every dot is a genetic variant, and the height of the dot is essentially the strength of the association signal. And if you're lucky and have a well-powered study of, I think this one's from inflammatory bowel disease, you find many, many strongly associated variants. And then the way that it's portrayed is the next natural thing is you look more closely at these associations. They pile up neatly under a gene, say the interleukin-23 receptor, and that gene fits nicely into a set of fairly well-understood biology that might lead you to think it's an interesting target, or that perhaps other genes in that pathway are interesting targets. And in fact, if I just take for a moment a disease I've worked on quite a lot, inflammatory bowel disease, the first part of that approach that I just told you about, identifying statistically robust association signals, has been incredibly successful. So from the time when GWAS really began in about 2006 to today, we've identified over 200 very confident, independent associations between genetic variants and risk of inflammatory bowel disease. So the question becomes, what biologically do those associations mean, and how do we feed them into target discovery? Well, we publish in the papers tables that look like this, that go on, in fact, for multiple pages, and we have lots and lots of these associations, and for all of them, we have information about where in the genome they are, which variant is the most likely associated variant, and indeed what gene is the gene we think is relevant in that bit of the genome. And sometimes we put the genes in bold because we have some slightly higher confidence in those genes. But the scam that is behind all of these is that, in fact, most of the time we have no earthly idea which gene is caused, or indeed even which variant is the right variant, because variation in the human genome is correlated from one nearby variant to the next. you actually see a signal that's kind of smeared out over a bit of the genome, and you really don't know which variant is causal, which gene it affects, and certainly not what the mechanism is that links the variant and the gene. And that was the situation that really brought the three founders of Open Targets together, who were Janet Thornton, who was the director at the time of the European Bioinformatics Institute, Patrick Vallance, who's the head of R&D at GSK, and Mike Stratton, who is the director of the Wellcome Trust Sanger Institute. And the idea was, can we set up a public-private initiative to transform drug discovery by systematically using the kind of data that I've just been talking about to identify and prioritize targets? And the motivation was that this is a problem that involves a wide range of expertise and really couldn't be done by any of these partners alone. And so in fact, we brought together the expertise of these different partners, and indeed, as of early this year, we added our fourth partner, Biogen, who are based right here in Boston, where Aaron works and is going to tell you a little bit more about later on. And the idea was that this particular aspect of the problem of understanding which are the good targets to go after has a large fraction that can be done in a pre-competitive collaborative way. So I'll just briefly say something about the two academic partners, EBI and Sanger, for those of you who may not be familiar with them. The European Bioinformatics Institute is Europe's, and indeed probably the world's, repository for bringing together, assembling, curating, interpreting, and presenting biodata. And there really is their mission to devote the resources necessary to, in the long term, support the collection and presentation of these data is, I think, very important. And they cover really the full gamut from genes, genomes, and genetic variation through the central dogma of expression in different parts of the body, protein levels, all the way to things like molecular structures, chemical biology, reactions, interactions, and it's based upon an accumulation and mining of the literature and other ontologies to relate these different concepts together. The Wellcome Trust Sanger Institute is the largest genome research institute in Europe. It led the UK's contribution to the Human Genome Project, and in the 20 years since has been focused on translating those activities into a better understanding of human biology. There are four main research areas at Sanger: human genetics, cancer and aging, which are of course linked by the process of somatic mutation; infection genomics, where we concentrate on understanding the genomes of both the host and the pathogen and how they affect susceptibility; and an increasing focus on cellular genetics as a model for some of these processes. And Sanger's research is built upon a set of high-throughput platforms that differentiate us, say, from a university department. And those are historically DNA pipelines and high-throughput sequencing, and also information technology, and more recently an increased focus on single-cell genomics technologies, as well as high-throughput cellular differentiation and phenotyping platforms. And I'll just say that we're also part of a wide range of national and international collaborations with other institutions about how to best use genetic variation data to improve human health. And Sanger and EBI together are on the Wellcome Genome Campus, just outside of Cambridge, England. There really creates, I think, a great atmosphere for these two distinct but quite tightly linked institutes that bring their expertise together. There's also, more recently, there's a conference center on the campus, as well as more recently the launch of the Biodata Innovation Center, which is a small incubator and spin-out space. So the campus is starting to grow from its purely academic roots to involve a wider range of activities. So what is Open Targets? How is Open Targets going to bring these different partners, private and public, together? Well, we try to base everything we do on a set of core principles that underpin both the bioinformatic and experimental research that we undertake. So first of all, as I mentioned, we believe that an important part of just understanding the biological mechanisms of complex disease and prioritizing which may be worthwhile as potential targets should be done in a pre-competitive way. It's too complicated a problem and it needs too many different types of individual for every company to try to build it in-house. And that's why we're extremely excited that both GSK and Biogen share this vision. They really believe this is something that's best done together. And different companies have different views on this, and I think that's totally reasonable. And we're just trying to set our particular direction based on this belief that at least the earliest part of this problem is done pre-competitively. And related to that, and consistent with the ethos of Sanger and EBI, we publish all of the results from all of the experiments we do. We write papers, we put the data in public databases, and indeed, the bioinformatic platform at targetvalidation.org is available to anyone in the world to use. And finally, as evidenced by the fact that we've already grown from 3 to 4 partners, we believe in a non-exclusive approach to building open targets and to finding other public and private entities who are interested in working together on this. So I just wanted to tell you briefly before I hand over to Aaron, who will give you a really nice kind of more detailed worked example of the way we work in the specific case of Alzheimer's disease to give you a bit of an overview. So as I mentioned, we have both a bioinformatic and an experimental component to Open Targets. The bioinformatic aspect is really focused on comprehensive and robust data integration. There are so many different types of data, the genetic association data that I started off with, gene expression data in specific tissues and disease processes, protein expression and function, being able to mine the literature, understanding existing interactions between drugs and targets. And many of these databases, in fact, are hosted at the EBI, but they don't necessarily do a great job of communicating to each other. There are many different gene identifiers that are used by the protein versus the gene expression community. It's not straightforward to ask a question uniformly across all of these resources. So the first thing we did was try to put together a single framework that uses a unified data model that Aaron's going to mention a bit more about, as well as try to develop statistical integration. So instead of just looking at web pages and trying to do your best to figure out how these things fit together, that we apply a model that gives some kind of a statistically motivated interpretation and joint score of all of the evidence that links a particular target to a particular disease. And I think it's actually already been a success even within EBI and Sanger that we've started to bring different teams to work together. So we have something like 50 full-time employees at Open Targets, but only about 15 of them sit in what we call the core group, and the remainder are all embedded in either EBI or Sanger teams. So when we need, for instance, access to the genome-wide association study catalog, we put one Open Targets person in that team and are able to leverage all of the work that they're doing to really get the best benefit for us. And as I mentioned, our main way of serving these data back to the community is through targetvalidation.org, which you can either access directly on the website, or if you're interested in building it into your own pipelines, we have an application programming interface which is open for use. So what does this look like? At the website, you can either say, I'm interested in a particular target or a particular disease. If you look at this example, for instance, it's the gene S1PR1. It shows these bubbles, which are different therapeutic areas, and within them, specific diseases where we have some evidence connecting this target. You can filter and sort the evidence of different kinds. If you're a geneticist like me and you only care about genetic evidence, you can look at that. You can drill down into any one of these associations. So for instance, between S1PR1 and multiple sclerosis. It'll show you what the evidence backing up that interaction is. And in fact, you can go all the way through, if you're interested, to the original databases. We don't want to become the database of record for any of these things because that problem has already been solved to some extent. But for instance, the example you just saw, clinicaltrials.gov, instead we're a portal and an integration to try to bring these different things together in a systematic way. You can also look at a particular disease and ask for a prioritized list of the targets that have different types of evidence. This is a work in progress. I mentioned we're trying to develop a score that essentially gives you a robust way of thinking about the different weights of evidence. That's a hard problem, and we're continuing to work on it. I would say the version we have in the current version 1.0 is our sort of best first effort, but we're really putting a lot of work into trying to improve that so we get much more specificity about the most valuable kinds of evidence versus those that don't contribute as much information. Complementing that bioinformatics approach, we have a set of responsive, dynamic, human cellular experiments. We try to be strategic in thinking about which experiments best fit within Open Targets. We want things where target information is the key outcome, where we can connect a particular disease and a particular target. There are lots of interesting problems about, say, characterizing the biology of a disease system. Those are important and interesting, but we're trying to keep our focus on these specific target versus disease relationships. And that involves inevitably a lot of genetics. We try to be genome scale where possible, and by that I mean we do a lot of genome-wide association studies and genome-wide CRISPR screens and things where we're trying to generate information about all the genes in the genome rather than a deep focus on one particular gene. And obviously it's critical if you're eventually going to make a drug to understand the biology of the target you pursue, but we don't want to replicate things which we think are done well in the companies and instead focus on our strengths, which is how can we profile the whole genome in functional ways and really use that information to not just help the starting point of our partners who might have been interested in one gene or pathway, but also provide for the future where serendipitously someone can discover something they didn't expect in those data. And of course, we want to build projects that leverage the technologies I mentioned at Sanger, the sequencing, gene editing with CRISPR-Cas9 technologies of various styles, as well as single cell and IPS technologies. And the way that that we sort of think about all of the projects we have ongoing, we fit them into this grid. So our three main therapeutic area foci are cancer, immunology, and neurodegeneration. We have some projects that cut across diseases. And we try to think about those projects in a few kind of scientific categories. One is human cellular experiments. So this is really trying to have models that have some physiological relevance to disease and are done in human cells. The other is we try to use, wherever possible, genetics as a tool. We believe the the important causal information about genetic associations is incredibly valuable in thinking about evidence that prioritizes targets, and Aaron's going to give some great examples of that. And then sometimes enabling resources that let us build the pieces that we need to do these other things. So just to give you a couple of examples before I hand over to Aaron, we do, as I mentioned, various genome-wide CRISPR screens, the most straightforward of which is in the collection of cancer cell lines that Sanger, the Broad, and many others have worked with. There are about 1,000 that have been profiled to death. The layer that Open Targets is adding is this CRISPR synthetic lethality screen. So essentially we're going to do up to 500 of the lines, do a screen where you knock out every gene, and you can see which genes, when knocked out, cause increased or decreased lethality in specific cell lines. Because of course, if you knock out essential genes, all the lines die, and that's not so interesting. What you're really keen to find are things where, in a subset of, say, colorectal lines with a particular genomic background, they're very susceptible to knocking out this gene. We're also trying to use the same kind of approach in other types of cells, things like, for instance, fluorescent sortable immune cell function screens. Those obviously require, I would say, a little bit more delicacy in getting the high-throughput CRISPR reagents working in cell types that aren't as amenable as some of the ones that the early work has been done in. And our hope is that we can tie these different aspects together to really link the genetic evidence, the possible follow-up of function of those genetically associated loci in human cellular screens, and that ultimately those will come back into the informatics portal so that it continues to improve and offer better data to scientists who are interested in building hypotheses from it. So with that, I will hand over to Aaron to talk about Alzheimer's.
So what I'm going to walk through is an example through Alzheimer's disease of how we're using genetics, genomics, and biomarkers to actually turn these variants that have been identified through GWAS into actual valid targets. So, from the outset, let's compare what makes a good drug target. What do we need to know about a target to advance it to lead ID? So, one, we know what the molecule needs to act against. We know the gene that we're going after. We have a therapeutic hypothesis about how we need to modulate it. Do we need an agonist? Do we need an antagonist? And what is that going to move in disease? And we have biomarkers available to access downstream pharmacology, right? But we can contrast that with what does a GWAS signal give us? Like Jeff said, we often don't know what the causal gene is under these GWAS peaks. It's really just a beacon saying there's something in the genome here that's important. We don't know the direction of effect of how this is affecting the disease. Is it increasing or decreasing something, and that's having the effect? And there's an unclear relationship with intermediate measurable biomarkers. So what can Open Targets do to transform GWAS loci into targets that we want to go after? Right? And so Jeff put a version of this figure up, but essentially GWAS and genetics focuses our target search. Right? We can eliminate a lot of the genome and the genes underneath those parts of the genome because the GWAS has told us, here's a beacon, here's something that's important about the disease. Now you just need to go and figure it out. And so in Open Targets, we've developed this evidence model. Right? So essentially, we want to be able to link all the genes in the genome and score them against every disease that we know about. And we build this link between targets and diseases through building evidence strings. And we do that through text mining, genome-wide association studies, expression studies, methylation studies. And we utilize the experimental factor ontology that's supported and developed at the EBI, which is essentially a graph structure that allows propagation of information to flow through the ontology. Right? And so in an intuitive sense, what the Open Targets evidence model is really doing is building up evidence strings, which individual therapeutic scientists within companies do, and essentially perform deductive reasoning on, right? They accumulate evidence and create a deductive reasoning string to say why a target is linked to disease. What we want to do is convert what scientists do intuitively through deductive reasoning into an actual statistical formalism. How do we convert what scientists do and what the human brain has evolved to do? How can we convert that into a statistical framework that allows us to do that very rapidly across all genes, across all diseases? And if anyone in the audience is familiar with graph theory or network theory, or lots of research has been done on Twitter and social networks, it's the idea of belief propagation. With a graph structure, we can propagate information as well as uncertainty down a path through a graph to get a sum total posterior probability of how strongly we can link a target to a disease, right? And the ontology is exactly this. It's a graph connecting pieces of information, and we can assign weights and the amount of information on each of those edges and essentially sum up the probabilities linking those things together. And we do this through linking genetic, genomic, and biomarker information, as well as leveraging all the drug information that's in Kimball into a weighted path from target to disease. And so I'm going to use Alzheimer's disease as an example to walk through what we would do for an individual target and how Open Targets is doing this on a genome-wide scale. So in 2015 came out the largest meta-analysis for Alzheimer's disease. This line here is considered genome-wide significant, such that anything above this line were very, very robust signal bound to replicate. What we can see is that there's a huge number of signals, 22 different genome-wide significant hits. And if we were just to choose this and say, all right, for any one of these loci, what is the information that we need to convert a GWAS locus to an actual target? Well, the first thing we need to identify is what is the actual causal variant underneath any one of those peaks. All right, and we can do that through statistical fine mapping using summary statistics or actually genetics. We can go out and do exome sequencing or whole genome sequencing and identify any rare coding variants in a particular gene underneath those peaks that are actually associated with a gene. And then we need to actually, once we know the variant, we need to say, well, which one of the genes in the region is that linked to? There we can also leverage rare coding variation. We can look at EQTL, expression quantitative trait loci, and identify, well, this causal variant that we found, does it actually change the expression of any of the genes in the region? If not, does it change the methylation landscape around any of the genes and therefore change its expression? We can do capture high C methods to actually ask, what parts of the genome does this causal variant actually interact with? Is it a nearby gene or is it several kb downstream? There are several pieces of evidence and papers that have come out recently, including Manolis-Kellis's FTO paper and the New England Journal of Medicine, showing that the FTO locus is one of these loci where sort of the causal variant and the peak has been known for a very long time, but what it actually interacts with are genes several kb away and is changing the way people are thinking about that locus for type 2 diabetes. And then to really turn a gene into a target, we need to have some sort of therapeutic hypothesis or a mechanism of action tying that gene to the trait. So is it associated with any intermediate traits or biomarkers that we know are associated with disease? And is the SNP or trait, SNP or gene actually associated with those biomarkers? So let's take a random locus, one of the 22 genome-wide significant hits. In the original paper, this was called the ABCA7 locus, simply because the top peak, the peak with the SNP with the lowest p-value, most significant p-value, is closest to this gene. But as you can see, this is an extremely gene-dense region. So, flip a coin, which one is the right gene here? So how do we transform this statistical genetic signal into an actual target? Well, over the last 3-4 years, there's been a raft of papers that have done whole exome sequencing and whole genome sequencing in Alzheimer's disease and have found a preponderance of evidence. SCAT is a particular statistical genetic algorithm to do burden analysis, and genome-wide significance for gene testing is a lot smaller than GWAS because there's a lot less tests. So this is a huge, huge statistically significant signal. And you can see that there are coding variants across almost the entire gene that strongly associate with Alzheimer's disease. All right, so this sort of put the nail in the coffin that put a very strong signpost that actually ABCA7 is probably the actual real gene in the region, right? And then we need to further identify this. What are the causal genes doing and what are the causal SNPs doing to that gene? So there's another statistical method called co-localization. It's a Bayesian framework for testing whether the signal that is causing an EQTL is actually the same signal causing disease association. This is an analysis performed by Carol Estrada and my group at Biogen, taking publicly available data and running this analysis, actually showing that the posterior probability that these SNPs are known to be EQTLs in the brain, and the posterior probability that the GWAS signal and the EQTL signal are the same signal is actually a posterior probability of 99%. Again, very strong evidence that ABCA7 is the actual gene in the locus. All right, so that gives us causal variance to causal gene, but we still don't know why would this gene be associated with Alzheimer's disease. So Barett et al. In 2009 has this figure of our current hypotheses around what's happening throughout the disease progression of Alzheimer's disease. And the amyloid hypothesis is still very strong, right? And also neurofibrillary tangles and tau is also emerging as a very important biology in Alzheimer's disease. So the question is, does ABCA7 and the SNPs that point to that gene, are they associated with any of these biomarkers that we track? All right, and this is just to show, this is a one way that we can display the ontology, but basically saying that here's Alzheimer's disease in the ontology. Currently in the ontology, it says it's a tauopathy. We're working on that to also make sure that it also is an amyloidopathy. But we know that beta amyloid through imaging is a biomarker for Alzheimer's disease, as well as many other putative biomarkers reported in the literature, right? So we can step through the graph and supply evidence and statistical information to try and make that link between Alzheimer's, a biomarker, and the genetics. So this is our recent publication talking about our phase 1b trial, testing the aducanumab antibody that reduces A-beta plaque in Alzheimer's disease. This is figure 2a showing that as you increase the amount of aducanumab given, the reduction in the amount of A-beta through imaging seen in their brains, as well as an improvement in cognition. All right, so now we have a biomarker that we can measure in a clinical trial, beta amyloid load through imaging. We have a therapeutic agent targeted against that is actually removing its target and showing improvement in the phenotype. So if we take the ADNI data, the AD Neuroimaging Initiative data, and we take their PET imaging data, do we have any evidence that ABCA7 and the SNPs that are causing the association to Alzheimer's disease, are they affecting this at all? So this, again, this is an analysis done by Carol Estrada and my group at Biogen. So if we take the ADNI data and we use their PET AV45 imaging data, this is one of the lead SNPs from the GWAS, the AD GWAS, and the A allele is the risk allele. And what we see is that this risk allele that is causing increased risk for AD is also increasing the amount of amyloid in AD patients' brains. So now we have a therapeutic hypothesis of how ABCA7 could possibly be linked to Alzheimer's disease. This is actually a pretty rare in European populations frameshift mutation that causes a truncation in the gene, which is actually fairly common in African Americans. And even though the sample size is slow because there's not a lot of African Americans in the ADNI data set, and this isn't super variant, but there's definitely a trending association of people that carry this frameshift have increased amyloid load. So now, what a therapeutic area scientist would do is essentially perform deductive reasoning with these pieces of evidence. All right, so we have a SNP that's associated with Alzheimer's disease through GWAS, through rare variant associations in EQTL and colocalization analysis. We know that the SNP is actually pointing to ABCA7. Therefore, stacking these evidence strings on top of each other, we can deduce that ABCA7 is an AD loci. Additionally, through clinical trial information with targets that target beta amyloid, we know that amyloid is associated with Alzheimer's disease, and by reducing, at least preliminary data, reducing A-beta accumulation in the brain reduces the effects of AD. And through GWAS of imaging data, we know that these SNPs that link ABCA7 actually shows that ABCA7 is influencing A-beta levels. Therefore, not only do we have a target, ABCA7, we also have a therapeutic hypothesis that ABCA7 is influencing beta amyloid levels in people's brains, which is associated with Alzheimer's disease. So we've taken this one example that one individual could go after. And what Open Targets and the Target Validation Platform does is actually formalize this in a statistical framework. So if you go to targetvalidation.org, type in ABCA7, what you see is that The scoring scale I left off, but it goes from very light blue to very dark blue, and the dark blue essentially says a posterior of 1, that this target is linked to this disease. And as you can see, the platform says, without a doubt, that Alzheimer's disease is the #1 indication for this disease. And this was all done without any human intervention through the statistical framework that we've set up. And so the power of the Open Targets model is that it leverages the expertise of all the different partner institutions. As Jeff said, EBI really is the world's leader in genomic data curation and storage. The Sanger Institute is a world-leading genome-scale biology institute. EBI and Sanger together also have faculty that are on the cutting edge of statistical method development and analysis. And the industry partners bring our experience and expertise in drug development and therapeutic area expertise to inform how we build these statistical models, how we integrate data, and how we present it to scientists. And all of our groups are fully integrated such that academics and industry scientists are working together on projects in real time, seeing data together, analyzing it together. And that the informatics and the statistical part are fully integrated with the experimental wet lab work, such that it's an iterative cycle of new results coming out of the wet lab, enhance and change the information within the platform. And that all of this is really rooted in human genetics and genomics, such that even our in vitro experiments are in human cells, not non-human cells. And really the idea is to turn the big data that genomics has produced over the last 15, 20 years into actual usable knowledge. So with that, I'd just like to thank the therapeutic area biologists in the neurology department that we've worked with at Biogen, Carol Estrada and my group, as well as the R&D, IT, and digital health, as well as the scientific leads from Open Targets that have helped develop this, and really the unsung heroes, which there are too many to list up here, but all the core development individuals part of Open Targets. And so with that, I'll hand it back to Jeff just to finish up.
Thanks, Aaron. Yeah, just to try to bring some of those things back together before we take a few questions, we're obviously not the only people who've had the realization that big-scale human genetics and genomics has an important role to play in trying to identify targets that are much more likely to ultimately be successful. And so I just wanted to finish with a few things that we think makes Open Targets unique and different from other efforts in the same space. And the first is that we try to put the user first in everything we do. That is, academics especially can have a tendency to build tools that do the things we want them to do, but what we really want here is to provide value to scientists working in the drug discovery industry. So we do that in a variety of ways. We have workshops where we show the early versions of the platform to scientists at GSK and Biogen, take their feedback, try to build iteratively a more and more valuable platform for them. And we have things like this scribbly bit at the bottom of the screen where each of those colors is feedback from a different user, and sometimes you get things like on the right-hand side in all capital letters, confused, which is good feedback because it means there's something we're not showing very obviously and we need to fix it. The second thing is that, as Aaron just alluded to, we really bring the partners together. So this isn't a collaboration where some amount of money is put in to do some science and then we'll fax you a copy of the paper when it's done. Instead, the project teams have industry science and academic scientists. There are Biogen and GSK employees sitting on the genome campus. There are some of our staff working in the research labs at the industry places. And it really is a different feel that different individuals are working together as part of these teams. And then finally, as we alluded to, this is we want to work at the stage that's at genome-wide scale, the genome-wide association studies, the whole genome CRISPR screens, et cetera. We want to provide these things that ultimately will hopefully be useful, not just for the target that got us started in one particular area of interest, but will continue to give more and more information more widely.